The science of today is the technology of tomorrow. A very good morning, one and all present here. I am Sandhya H. First MSc Biochemistry, Mangaluru University. Here is a one more day to learn and inquiring a mind. On this beautiful occasion, I would like to welcome Dr. Nagaraj Nagathihalli, Assistant and Director for Basic Science Research and Associate Professor of Surgery Research and Sylvester Pancreatic Cancer Research Institution, University of Miami, USA. And also I would like to welcome Professor Chandrasekhar G. Joshi, Coordinator of MSc Biochemistry Program, Mangaluru University and Coordinator of GIAN Program. Welcome you, sir. And also I would like to welcome the faculty members of Department of Biochemistry, and also I would like to welcome all the enthusiastic participants for this beautiful GIAN workshop. Once again, I welcome you all. Have a nice day. Thank you.
So good morning, everyone. I hope you had a nice last 3 days. So there are 2 more days left. If you are exhausted or you're still enthusiasm to learn more. Okay. Today we're gonna have. I think we keep talking about natural compound. I think there are many of you are interested in natural compounds and screening, understanding how to conduct experiments, and also how to study natural compounds. Even from the plant extract, there are people talking about plant extract, and after the extract and what you should do, how do you conduct the research and stuff. Maybe some of those aspects of extraction and details, I may not go in detail, but what is important is that what you should do once you have your compound, or once you think that there's an active compound exists. That's a completely chemistry part, that you have to collaborate with someone, with the chemist, understand that that compound that what you are actually derived has the ability of doing something, right? So I'm gonna tell you a little bit of story so that you'll understand the story of how we did it and what we did it, and the details of it, so that probably it will help you and in your experiment and your research how you should conduct based on some of these parameters that we have done, right? So first few slides, you will see the general natural compound study and what's going on, and then later on we'll skip to our specific natural compound details and the work. So these natural compounds are organic compounds that are produced by living organisms, including plants, microorganisms, animals, and marine species. Like some of you are working on this plant extracts. So these natural compounds have a rich history, right, of contributing to new therapeutic medicines. Also, they have this. They've evolved this structurally to fulfill specific biological functions, right? And also typically exhibit a broader chemical, diversity, characterized by a high number of these sp3 carbon atoms and chiral centers and all those things. So these are all chemistry. That's why you need to really collaborate with chemists if you don't have the experience, right? The classical nanoparticles, right, that led to the discovery of these drug molecules are for therapeutic use. You can see some of these natural compounds, they were already in the therapeutic use. They're all derived from the natural compounds, and they're all approved from FDA or clinically from approved. For example, here morphine, which is actually painkiller. You know very well that morphine, when a patient have pain, pancreatitis also, I think people give, doctors will give morphine. So I think it's important. So penicillin, it is well known, right, is also from natural compounds. So what I'm trying to tell you is that most of the approved drugs, right, maybe 30 to 40% are from the natural compounds. So it is important to study. So as an Indian rich medicinal plants, and even in China and Asia is rich in medicinal plants and medicinal. We have all the history of and how the Rishi Maharshis were using all these natural products for therapeutic purpose. Even Raj Maharaj's time also. So these are all true. They were using the really natural products, natural plants, extracts to therapeutically they were using. It is true. It is just that you have to think scientifically today in this today's world. How are you gonna study those natural products in a therapeutic purpose? But you have to do it right. You have to conduct the research correctly and right, so that it will be useful to the mankind, right? So these are some of the anti-cancer agents. They were approved, and they are from the natural compounds. Okay, you can actually see some of the STAT3 inhibitors which we used it, and there are a lot of other. Just to mention few anti-cancer drugs that were from the natural compound. Remember, I'm going to show you my own example, but before that, I want to show you some of these compounds are really, if you go to see the literature, it's a recent literature from last year. Actually, they have done great job. You should read this paper and see what they have inside all the natural compound history and what exactly happened and all, if you are studying natural compounds or want to study from the plant extracts. Okay. The rationale for using the natural compounds in cancer interception. See, there are structural diversity and novel mechanisms. That, for example, if you take the diverse chemical scaffolds, these natural compounds often possess complex, evolutionary optimized structures that are less common or absent in synthetic chemical libraries. That is a advantage, right? This diversity expands the range of potential drug targets and mechanisms of action for cancer interception. Also, they have established clinical precedents. That means that historically, many anti-cancer drugs, for example, you can take, I was talking about paclitaxel yesterday, right, in my study, in the chemotherapeutic study, this research and make sure they are doing what you are thinking that can be used in therapeutic purpose. So multi-target target potential. So for example, many natural compounds can modulate multiple cellular pathways implicating carcinogenesis. So today you are lucky. So I'm gonna talk about exactly that multifaceted, multi-signaling pathways that our compound, how that happened in our study. Right? Basically, we're going to hit that study. So these natural compounds may exert the chemoprevention. Okay, the people have studied. So some Indians, many Indians, actually when they go outside abroad and all, they usually do a lot of chemoprevention studies. Something them naturally, ours, probably you all doing the plant extract and natural compounds. Even I did it too. So we have conducted also. So it's like, probably it's a birthright for us. Probably we all do the chemoprevention studies and natural compound. We think about it. Don't know why, but it's just coming to us. Probably we keep doing it. I've seen many Indians working in outside the world, all over the world, in many countries. They're all working on chemoprevention studies. Many established investigators and many investigations have come from the natural compounds. And even the turmeric. Many people have done and worked on turmeric and things like that, right? So when you come to cancer prevention and interception needs, right? You have these early intervention studies by targeting either pre-malignant disease or early stage cancer. I showed you from the pre-malignant, normal to pre-malignant to pan in 1, 2, 3, and then the cancer, like that in many cancer, many progression studies. So early on and in between or in the later stage. So, there are many pathways. I showed you many signaling pathways in the early stage and the late stage. There are many signaling pathways changes, right? You need to know all this, biology and signaling before you take some compound and hit it. Because you need to know this compound where it's going to hit, but you need to screen those signaling pathway. That is what I think you're going to see today when I show you my work, right? See, these natural compounds may exert the prevention activity, through anti-inflammatory, also anti-proliferative or anti-apoptotic effects. Then we discuss about the low toxicity profiles, are important, right? When you are studying chemoprevention or the natural compounds, you have to think about the toxicity. So these compounds have a low toxicity. That's why they get approved easily when compared to a synthetic one. That's the advantage a lot of people study on these things, right? So the key requirement for preventive agents in minimal toxicity, right? Everyone in every company, those who have synthesized some compounds, all suffer from this toxicity issue. So, but these natural compounds can often well tolerated. So they do well tolerate because it's natural, and making them promising candidates for long-term administration in a high-risk population. For example, if they're very age-old people, when they have a cancer and high-risk range, so this natural compound is much better than the chemotherapy, other synthetic one. There are some chemopreventive agents are from natural compounds too. They do use, okay. And then comes the technological and methodological advances. So we use one of the omics guided discovery. So it's important to understand what is omics, and then those technology. I think we touched base yesterday a little bit here and there, but there is a advance in the metabolomics study, proteomics study, transcriptomics studies. And all this will help identify the mechanism of action of these natural compounds. You have to sometimes combine all these omics study. This is study I'm talking about. So investigation is something different and studying is something different. So I'm talking about studying, screening, and looking at the proteins, looking at those genes, what they change. Remember one thing. So for example, you hit with a natural compound, you take plant extract and study. It may change the protein. It may not change some genes related to that protein. Sometimes only change in the gene level may not on the protein level. Things can happen. Okay, I have seen. Sometimes it will hit the really transcription level. That's why they do the omics study. Means that they do the proteomics, genomics, and all the kind of metabolomics pathways and studies. The reason is that we don't know where it is hitting and what it is hitting, whether it's a gene or the protein or some enzymes or pathways and things like that. So also it's, I think technologically it's important to improve the isolation and structural elucidation. So whenever you have a natural compound, when you isolate it and you have to you think that your isolation method is perfect, may not be, because remember our study was not perfect too. When we evolved understanding what's going on in the whole area, and then we learn more and more, and then we isolate better and better, right? So you get the compound, because probably 10 years ago the isolation methods are different, and today the isolation methods are different, because there are a lot of things that they learn. We learn in the research that what is right and what is not right, right? It's okay what you have done 10 years before, and today if you see those isolation methods may be different. So you need to learn those isolation methods. Don't follow the procedure 10 years ago, what the PhD student, someone senior is conducting and doing it. So you have to upgrade and update yourself, right? And then conduct your experiments based on the present, what's in the world, what they are, other laboratories in the other world they are doing. Because when you submit your paper, when you put the isolation method, believe me, it happened to us too. I'm just letting you know early on when we started studying. So they will reject based on the way that you've done the isolation methods. So you need to really upgrade yourself, that up to date yourself about the methodology, isolation methods, right? Or collaborate with someone who knows all these things, or who is actually updated themselves in other laboratories, right? So that's important. So there are many technologies evolved, right? NMR and micro ED. Nowadays they talk about micro ED, microcrystal electron diffraction studies. So mass spectrometry, MS, you have to do. You all know, if you are doing the chemistry and the isolation, you have to use the mass spectrometry study. and they all accelerate the structural identification and even of the molecule available in the minute amounts if you want to study. But your purity of your compound is a key thing. Whenever you wanted to study the purity, you have to always think about the purity. Now for that you have to use the technology to make sure that the compound that you're hitting, and that is what I think is naturally is coming out. It is not something artifact, right? That structurally, anyway, you will know when you start synthesizing those compounds, right? Okay. When it comes to methodology for screening the natural compounds, so there are a lot of we talked about crude extracts versus fractionated samples. So traditional workflow begin with the crude extracts, which then undergo fractionation to isolate these active molecules. So you have to find the active molecule. There may be many, right? We can give you examples. One, if you think that one compound, you say for example, one HA compound something. So then there is HA 1, 2, 3, 4, 5, because there are many many of them in when you isolate. But you need to find the one which is active. Probably not all of them are active. So how do you do that activity assay? And these are all something that you have to collaborate if you don't know. And then this fractionation, they undergo to isolate active molecules. However, the pre-fractionated libraries can expedite this process by offering semi-purified fractions enriched for drug-like properties. For example, moderate polarity and improved some solubility, things like that. You can actually make it. You can actually make the drug-like properties that you can conduct those experiments and see whether these compounds have the drug-like properties or not, right? And also reducing the complexity. This fractionation enhances the chances of detecting unique hits. It also avoids some of this interference from complex mixtures, and also we can actually streamline the follow-up isolation of these active compounds. So when it comes to high throughput screening, HTS platform, there are many ways that you can conduct. There are multiple ways that you can conduct high throughput screening. One is the common way that they screen is a cell-free biochemical assays, like target specific binding or enzymatic activity tests, like we do the kinase assay, like epigenetic targets that we talk about, so those kind of things. And there are cell-based assays, they call phenotypic screens. It's like we all do in the laboratory. For example, you all do the viability assays and apoptosis, and also use sometime maybe imaging for a broader explanation of this compound activity. And also there are some short-term in vivo models. Like for example, you can say wherever it's applicable, you can use some small in vivo study. It's okay, but you don't have to be genetic model or hi-fi technology. You just simple in vivo experiments can also be useful, like or maybe a flank injection of some of these cells and study your compound, things like that. It's okay. It's just about just to see whether it's going to work or not for you. Okay. And people use for the toxicity efficacy study, zebrafish study, and invertebrates they use. There are a lot of technologies evolved. For example, zebrafish study is one of the great and collaboratively you have to collaborate with someone like Vemula. Or Vemula is my collaborator. What I'm saying is like that. There are a lot of other chemists all over. There were like from Kerala, from Maharashtra. I've seen many of them in some other conferences in US, and when they came to ASCR. And I go see sometimes when they bring some natural compound chemist, oh, I'm chemist, I isolated this, I wanted to come to this conference, I'm here. So they're great, I mean, like that. And I've seen myself, so many of coming from different places and presenting their work there. And when I saw that, oh my God, it's like, there's a lot of them. Like that, you have to search these people and collaborate with them. So collaboration is not easy, right? You have to have the money, you have to have the resources, then only people come. Otherwise, you have to have an idea and knowledge, and also same time you have to publish. Publication sometimes is easy for you if you have a knowledge, awareness, lot of things. But we feel always that we have to generate data. I keep telling from the last 3-4 days, because I cannot tell you exactly what to do, but I can tell you that with experience that we have been through, and you can actually do it. Okay. Overall, that this is something promising target. What I'm not telling you, a lot of other things that we have conducted. And what tells us that urethane can do, think about it. If you sit back and think about it, you yourself will get an idea what I can do yourself. Actually, like that, many investigators have taken urolithin A. Today, urolithin A work, if you Google it, PubMed, tons of papers came out, Nature, Science, everywhere. I'm not kidding. Even though they have done the mitochondrial health, and they have done many other ways. I have done only in the pancreas. And my pancreas issue, because I cannot do so much, right? I can only do a little bit, but at least my focused on what I am supposed to do. There is a focus there. That's very important. Okay, you cannot do everything. You cannot be master of everything, right? You focus on what disease or what you want to do, and focus on it, and I think you will reach the goal. And like we have conducted all these things. Reprogramming, toxicity profiling, and intervention studies and chemotherapeutic studies and all those things. So there are a lot of future, like preclinical studies, microbiome investigation. A lot of things are going on in the laboratory. But important, I think, overall, learning from it, what you want to do, how you want. Sorry. So there are papers published. Like for example, if you see this paper, there are many investigators working in many different areas. Kidney, you take liver, intestine, immune system, joints, brain, heart, muscle, whole organ. There are many, I see a lot of them working on it because that compound worked so much for us. Okay, the same way many can synthesize themselves. Once they know the structure, they can synthesize and conduct those experiments. Now even they sell in Amazon, you can actually buy urolithin A because it helps gut. It helps if you have a gut problem or issue, an inflammation issue or something. The urolithin A helps. That's why it's becoming famous. So that's the story. Those are references. I'm happy to take questions if you have one or two. Yeah, go ahead. You have to use the mic.
Hello. So the problem with all the natural compounds is its bioavailability. Did you find any issues with the bioavailability of the drug?
So we did not, because the urolithin A is a natural compound. It actually synthesized in the gut and metabolized in the gut, and it's a metabolite. So that's natural. So basically, the bioavailability is very high. So no toxicity at all. But see, anything too much is too bad, right? You cannot take too much because, oh, urolithin A is natural and bioavailability is high, let's take more. No, it is actually lead to another problem. But what I'm saying is that naturally, if you take natural compound a little bit higher than even the synthetic compound, it's okay. It actually fine, but you cannot take too much. Okay.
And also, sir, I just noticed you had used PI3K inhibitor. Why didn't you use directly AKT inhibitor itself?
Yeah, we can use any of them. We just use the clinically approved drug. So the one we feel good. It's not that is the only drug we used, right? We screened a couple of other drugs. We only showing the one actually, fantastically worked, and naturally comparing to that. It's about that. Research is like that. What you see, is only the outcome of so much of work that you conducted in the laboratory, right? Yeah. Good questions. Thank you all. You have a question? Okay. Okay.
Since it's urolithin, it's not affecting the HPMA, that's normal cell.
Yeah.
So do you think that there is a lack of receptor on those cells?
So we did not go through and check anything because at that time all we wanted is a therapeutically we want to see whether it works or not. That's our first work, and we did not look at the therapeutically like what whether they present any receptor or anything like that.
So
On that the work.
is it a hydrophobic or a hydrophilic molecule?
The urolithin A.
Urolithin.
Yeah, it actually worked the same way.
Is it hydrophilic or?
Yeah, hydrophilic.
Hydrophilic. Okay. I thought there might not be receptors on the normal cell line because many a times, so cancer cells, sometimes they overexpress some of the receptors.
Remember this urolithin A is actually, if you take the AHR signaling pathway, it actually activates through AHR signaling pathway, right, in the gut, right? So the binding on the AHR signaling, right? It may be the binding affinity of this urolithin A is important. So what we have not done is that looking at the gut receptors, because we were interested in the pancreas, so we did not look at the receptors. Because we did not look at the AHR signaling on the pancreas. There were paper published on the AHR signaling on the gut, and they worked on the receptor and the ligand, urolithin A, how it binds and affinity towards the AHR signaling and all those things. So we haven't looked at those kind of things.
And one more thing regarding the dosage of this urolithin as well as PA3 inhibitors. So this has been used in micromolar and that has been used in different doses.
Correct.
When you
correct.
study.
So the one that clinically approved, it's fine because we know the toxicity of that clinically approved, it's published paper, and we know how much we have supposed to use. The natural compound urolithin A, we used whatever the amount we used is actually fine completely. Even though you can go actually higher dose, nothing has happened. Toxicity level was very low. So that's the reason that we went down because we wanted to do the immunotherapy to it, and also we were doing the chemotherapy to it. We just don't want to put a lot of urolithin A because we are combining with other compounds later on. So we were just using the lower dosage when compared to naturally. So how much to use? Okay, thank you. I think we have a tea break now. Thank you so much, everyone. So we can discuss. If you have any questions, we can have a discussion. Thank you.
Good morning. I hope you are refreshed now, and it is a fourth day, and I hope you are enjoying the journey. And we came across so many topics related with the in vitro as well as in vivo studies, and starting from the natural compounds as well as the mechanism of the action of the molecule, and in future also we are going to study so many other aspects. And whenever you are studying all these molecules or basic study, if you have to do, we are using the cells for our studies, and that's usually we go for cell culture techniques. In this tutorial we are having few questions, and then we will I sent you already the study material today morning, and the question start with what is actually the cell culture. Cell culture is basically it is growing the cells maybe out of the living organism. You are taking out the cells, and in a controlled atmosphere you will maintain the pH, you maintain the conditions which are existing in our body to we mimic the same condition outside in the laboratory. That we call it as tissue culture or cell culture. Actually it started in long back, and that time when it was started, actually the scientists, they were growing in the beginning these frog embryos and all. It was they were using those the cells majorly because these cells or the organisms are actually the cold-blooded animals. So you need not to provide them the incubation or temperature and all. So they were thinking like that, and later on the study went on, and they started making use of different media. Earlier they used to take some extracts of the living organism, and in that one they used to place the tissues, because initially this study was majorly related with the tissues, and most of the occasion overlappingly we use the terminology tissue culture and the cell culture. But that tissue culture, whatever it was done long back, the same terminology we are using now also. But the way we are doing the cell culture, from maybe compared to earlier days, it is totally different. We have a definite media, we have a definite protocol, we do have so many aspects that has been changed. And the other question was, what is the good laboratory practice, one thing, and what are the minimum requirements for a cell culture laboratory? Because being the students of this, maybe the cell culture or any other technique, first of all, when we go to the laboratory or if we are designing the experiment, we should know what do we require. That is the basic thing. And next thing is that what we should do and what we should not do. That we consider it as the good laboratory practice. Because there are certain things that should not be done in the laboratory, and there are certain protocols that should be followed must. That we call it as good laboratory practice. And here minimum requirements you need, as you know, when you go to the cell culture lab, you come across with, and if you see any laboratory, you find that one thing that is common in all the cell culture lab is biosafety cabinets. That is actually the place where you are doing the handling the sterile material, including the cells. And along with that one, you need even cell culture media. So in order to maintain the cells, you need certain media or the growth, maybe the one which supports the growth of the cells. If you are not providing the nutrients properly to the cells, they may not able to grow properly. So we have to discuss about the different media. And another one is that regularly we are using phosphate buffer saline, we will see later. And what happens is that there are certain cells, they actually adhere to the surface of the flask or surface, because once they attach to the surface, we are supposed to remove them, because if they grow in number, which is if they reach certain number of cells, we are supposed to subculture them, just like what you do in microbiology. You are continuously maintaining the culture for long time by subculturing. Here we are also subculturing this the animal cells by using, because we have to detach if it is an adherent cell which is attached to the supporting material, then we are using trypsin EDTA, and later we need even centrifuge and centrifuge tubes, because sometimes you need to centrifuge and concentrate the cells if you have to go for further seeding. And later on, whenever you are doing certain experiments, you need to know how many cells you have seeded in the tube. In such condition, you should have a knowledge about how many cells are present in that medium. So in order to do that one, better we should have hemocytometer, or sometimes if the lab is well funded, the better we can go for the automated counters. And even we need flasks, maybe for seeding the cells. And what should be the laboratory setup, because along with that one I can tell what and all that is needed for the basic cell culture lab. Here we need the dedicated space, especially for doing the sterile work, where you are using the separate chamber or separate room for handling the cells. Here what we will do, we have a lab, suppose say for example if I have this room and I cannot use completely, because if my space is limited, I cannot use everything for the cell culture. Better thing is that you make a partition so that you can avoid so many contamination in that area. So by partitioning that area from the other area, because you are not only doing the cell culture, while doing the cell culture also you are doing various other works. You are supposed to prepare the media. Suppose if you have not purchased the prepared media, you are supposed to prepare the media, or you may have to wash the glasswares, or you may have to keep other chemicals, or you may have to keep the antibodies, or you may have to store the media and all. So for that and all you need a space. Everything cannot be clumped at one region in the cell culture laboratory. So what you have to do, better you keep the space separate for the cell culture, and that area let it be separate from all other activities which you are doing. And sometimes maybe you make a partition in such a way that whenever those who are having the work in that area only will be there in that particular area. Otherwise what happens is that there is a chance of the contamination, because you are going from outside and there is a chance of taking with us, maybe there are contaminants, and that may affect your studies. So you have to be careful about that one. And one more thing is that you need the controlled environment. You are supposed to maintain the temperature so that the media is not disturbed, or even the cells are not disturbed, and you may have to use a proper environment. And work zone, what way you maintain the work zone? You need a clean area, that is for storage of your media and the reagents or equipments, and where exactly you are doing your culture work, that is biosafety cabinet, it should be very clean. So that, because why I am telling all those things, because you think that you have done a very good job, but you don't know what are the contaminants you are introducing when you are doing the work. Because there are certain cells, even if you are handling, they may actually cross contaminate the other cells. This is what happens especially with HeLa cell lines. So this was actually one of the oldest cell lines, that is immortal cell lines, that is maintained. It was in 1951, it was taken from Henrietta Lacks, a cervical cancer patient in Johns Hopkins University, and it is growing, and the number of cells that are used in the lab is more than her size itself. So when she died, she was around 31 years age, and nearly 50 to 70 years, so many this cells, or so many times we have used the cells, and different cell suppliers, they are giving the cells. We maybe many people they study or use that one for various cellular maybe cell related activities. The major problem with this HeLa cell line, because I am taking one example and it will be easy to understand. These HeLa cell lines, even they can be, they can cross contaminate and dominate other cells, and they can grow in that one, and they prevent the growth of the cells which you are interested. And they can even actually transmit through the air also. That's what some studies says that. So in such occasion, suppose if that is the case, if there is a one aggressive cell, and which even if it gets transmitted or contaminates through the air droplets, then your study is gone. You will not know whether you are growing the HeLa cell line or you are growing some other cell line. So in that case, if you don't maintain your culture area properly, then there will be problem. That's why we need to maintain always this culture area that should be properly maintained, and it should be neat. And once you got a clean area, you need to maybe incubate the cells. Cells, they have to grow in the media. So if they have to grow in the media, we are supposed to give them proper temperature and the conditions which favors the growth of the cells. Therefore, that one we need the incubator, and at the same time you need even waste disposal area, because whatever I am telling, you can observe in the cell culture lab whether we do have that kind of facility. Because usually when we go, if we don't have the knowledge about that one, that's why we kept this tutorial first, so that you are sensitized to the things what it is done at the cell culture lab, and you can see what and all the way that has been maintained, and why they have certain restriction when you are entering into the cell culture lab. So along with that, as I mentioned, let me tell about the biosafety cabinet also. So biosafety cabinets, actually it is necessary for aseptic handling of the cells, because you don't want anything to contaminate your cells, and you are running a costly show. By chance, if there is a small contamination, and if you are not able to predict whether it is right or wrong, it may affect your results, and you will not have the reproducibility of the results. So for that one, we need type 2 A2 biosafety cabinet, because that is considered to be the one which regularly we are using for the cell culture studies. And not only that one, so one thing is that we need a separate area. Second thing is that we need the biosafety cabinets, we need the incubator, and we need the inverted microscope where we have to observe these cells. So cells can be observed, or morphology of the cells can be observed using these inverted microscope, because the cells contain the media. You cannot take the normal microscope, usually what we are using, that is upside down. It may not give you the clear picture of the cell. So suppose if you are observing from below, you will get a clear picture of that cells. That is what actually inverted microscope is. So that we will use. And sometimes you have to, because you have to seed the cell in the sense you take a stock of the cells, and or if you get cell from cell repositories like NCCS Pune, you are supposed to store them or you are supposed to seed them. Seeding in the sense you are growing them. When you have stored that cells for long time at minus 196 degrees Celsius, that is the liquid nitrogen can, so it is at maybe actually it is straight away you cannot use the culture, or you have to thaw that one. So it should come to the kind of the 37 degrees Celsius. You immediately thaw those the vials or ampules at 37 degrees Celsius, and after that you will seed into the flask that contains the media. And there you need the water bath to maintain uniform temperature, or even autoclaves, because if you have to sterilize anything, you need the autoclaves, and refrigerators and freezers. And there are different kinds of refrigerators maybe that are maintained at 4 degrees Celsius, and there are the one which are maintained at minus 20, and there are certain freezers, they are maintained at minus 80. All these things we are using for storing different the chemicals or solutions based on the necessity or based on the conditions. And as I told, liquid nitrogen is used for storing the cells. Usually whenever you are storing the cells for the long time, you need something which prevents the distortion or maybe the deformity or even the spoilage of the cells if you are storing for a long time. So in that condition, maybe that case you can go for the very low temperature, that is minus 196 degrees Celsius, and along with that even you need a pH meter. Suppose if you are setting the pH, usually nowadays nobody goes for the preparation of the media. Earlier days they used to take the powders, and that powder used to be dissolved in the big container or small container, whatever you need, and it used to be filtered, and then it used to be used. It was used to be used. But the thing is that there is a lot of chance of contamination, and nowadays as many prepared medias from the suppliers they are available, instead of working so much on those wasting time on the preparation of the media, you can straight away purchase that the prepared media which is already it is present in a aseptic condition, and you can use that one. It will be much easier. Or if you have the facility to prepare the media, that is fine. Or even osmolarity can be checked. Suppose if you have osmometer, and these are all actually the basic materials that are required or equipments. And along with that even we need certain consumables as well as the accessories for our studies. So we need tea flasks, and there are different sized tea flasks they are available to maintain the adherent cell. Here those who are not familiar, just I mentioned what are adherent cell and what are suspension cell. So adherent cell is the one, if you take the cells and if you grow in a proper surface, they come and bind to the surface and they fix to the surface, we call them as adherent cell. And those which are actually present in the suspension, they won't come and bind to the surface, solid surface, we call them as suspension culture. Okay, those who are studying biochemistry or maybe others, they are familiar, but there are some people from the chemistry, they may not be familiar, that's why I am telling about that one. And you need even petri dishes, maybe if you are growing the suspension cultures or maybe any studies if you have to do, there are different petri dishes we are using. So there are different sized petri dishes are also available, like 35 mm or 50 mm or even 90 mm, depending, or even bigger than that one that are there. Usually we go up to 90 mm when you are going for the cell culture, but rarely even the bigger one they are used. And multi-well plates, so usually either it is a 6 well plates, or even 24 or 96 well plates can be used, or as Dr. Nag was telling, even higher number of the wells they are available, so that can be taken, multi-well plates. And spinner flask, that is majorly used for suspension cultures. And whenever we are using or handling the cells, usually we go for the pipetters. Earlier days, usually they used to use serological pipettes. Whenever I was doing the PhD, I used to use that serological pipettes, and we used to keep on a bulb, and then you have to transfer that media and cell using that serological pipettes. But nowadays, the automated pipettes, they are available, and if you are using these microplates, that is containing 96 well plates, even multi-channel pipettes, they are available, and even the automated pipetters, they are available, so that depending on your fund availability, you can plan and you can study, or you can make use of this one. And even the micro pipettes, suppose if you have to add say for example very low quantity, so you can use that one. So serological pipettes, usually we use for 1 ml or 5 ml, 10 ml, even 25 ml we use serological pipettes. But if the volume is less, maybe there are pipettes, they are available, even that micro pipette we say, or pipette men. So even for 25 ml we do have, 5 ml we do have, or even the smaller volumes like 0.1 microliter, all those things. Depending on the necessity, you should plan what type of this the pipettes you are supposed to use. And then what is the other essential commodity, or maybe the material that is required for the cell culture lab? The major thing is that we do need the media, different medias that are regularly used for growing the cells. The major medias that include are basic medias like DMEM, Dulbecus modified essential medium, or RPMI, or minimum essential mediums. And along with that, even we do use the FBS, or fetal bovine serum, or sometimes even serum free media is used. So because sometimes maybe that depends on the cell, that's what I am telling. It depends on what study you are going to do. Accordingly you should select the media, and accordingly you should select the flask, or accordingly your maybe the other materials that are used. And we need to remove the cells or detach the cells from the attached surface. We are using trypsin EDTA, phosphate buffer saline, usually we are using for washing the cells, because sometimes if the cells are to be detached, and if you are adding the trypsin, and if you are not properly washing that the cells, usually trypsin may not act, because the serum might contain trypsin inhibitors. And sometimes we need to add even antibiotics, because we want to take care of the cells that should not be contaminated from the microorganisms. So in order to do that one, what we will do in the media, we will add antifungal and antibacterial the chemicals like penicillin and streptomycin combination we will use, so that we can avoid the most of the microbial contamination. But there are certain contamination, they go beyond even the microbial contamination, that we will see later. And even plastic waste, whatever we need, or even glass bottles, media storage. And I am actually clubbing these two materials and the good laboratory practices. Okay. So whenever you go to the lab, you should know what you are doing and why you are doing. And maybe this, if you are going to the cell culture lab, better is that you wear a lab coat, so that you are not taking any contaminants from outside. Okay. cells are sensitive to take up that contaminants, better you avoid all those things by the dedicated lab coats so that you can avoid most of the contaminations. So usually the country like us, we have a dust problem. Maybe that dust may affect your culture, so better you take care of that one. And once you are handling the or once you are working in the hood or biosafety cabinet, better wear that one. Or suppose if you have to talk, or better you should not talk when you are working in the this biosafety cabinet. Make sure that it is covered with a sheet so that you will not spill anything on the media you are working. And make sure that UV sterilization of that area, because that is the best way you can avoid most of the contaminations. So here even the biosafety cabinets, they come with UV sterilization. Best way is that you swab that the biosafety cabinet before work and after the work. And before work you switch on the UV maybe for 15 to 20 minutes and switch on the UV after the work also for 15 to 20 minutes. So that is the good way you can avoid most of the contaminations. And moreover, you are making a way for the other person to work on that. Maybe whenever too many people they are working, there is a chance of crowding. So that may affect your work. So try to make your schedule accordingly. So whenever you are having too many people, you are having work, you fix the time. So that if you have the work, no one else will be, maybe they need not to expect you to finish off faster or something like that. You take your own time and finish off the work. And when you are doing something hurriedly, there is a chance of making mistakes. So you avoid such things by making sure that you are having a proper time, you schedule a proper time, and then you work. And decontamination protocols. Not only working is important, after that how you decontaminate the things what you have used. Regularly we use, suppose if you have to work in the biosafety cabinet or any culture work, better you swab your hands with the 70% alcohol. So that avoids most of your the contamination from your side. Okay, it has been shown that even the cells that are dead cells present on us, it may transmit it if you use the bare hand. Suppose if you are wearing the gloves, better you wipe once with even 70% alcohol. You can avoid, maybe by chance, if there is any chance, you are avoiding. But ultimately, the thing is that you should avoid the contamination. And biohazard material should be thrown or maybe disposed at a proper place. They should be designated with biohazard symbols. And most important thing is that whatever you do, you keep the record. So unless you keep the record, you don't know what you are doing. Suppose you go to the cell culture lab, you did something and you don't know what you did on that day. Next day you will be repeating the same thing. You did not know what you have done in the previous day. So then it is a repetition of the work. And moreover, you will not recognize what are the mistakes you are doing when you are working with the cells. So try to figure out what actually you are doing and what are the challenges you are facing when you are, because initially when you are starting the cell culture, you are new to the technique, you are new to the work. When you are new to the work, always make sure that you are maybe following a proper protocols so that even others they can work smoothly in the lab. You maintain a proper lab notebook, that is one of the major aspect, and you maintain inventory management systems. You maintain how much consumables you have, how much stocks you have, so that your planning the experiment will be easier. And you maintain certain standard operating procedures. So that SOP will help you move in a proper direction. And the other question was that what is the role of carbon dioxide incubator in cell culture? So here we usually use carbon dioxide incubator for most of the cells. So carbon dioxide incubators, earlier they used to provide the temperature and carbon dioxide that used to be supplied in that chamber. It maintains the humidified condition so that the media will not get dried. Okay. So then here nowadays even the chambers that are available, they are coming up with various adjustments, like you can sterilize that the carbon dioxide incubator by increasing the temperature inside that the chamber. Earlier you were supposed to the cleaning and maintaining the sterilization was a big challenge. Now even the suppliers, they are designing in such a way that they are built in with certain kinds of sterilization. Suppose you can take the temperature up to 180 degrees Celsius and you can make sure that your carbon dioxide incubator is free from the any contaminants. Okay. And at the same time, whenever you are using the carbon dioxide incubator, usually you fix the carbon dioxide around 5%, and carbon dioxide present in the chamber, it diffuses into the media, and it may interact with the media to form that, you know, the reactions. Those who have studied biology, it becomes carbonic acid, and it becomes bicarbonate and H plus. So it will help in the proper maintenance of the medium. So whenever you are using the carbon dioxide incubator, there are certain precautions you need to take care of. The major thing is that because all of us, we will be keeping in the same incubator, we cannot have 10 incubator at a time. So whenever you are keeping certain cells for incubation or any maybe majorly the cells, make sure that the whole the area inside is clean, and regularly make sure that area is kept clean always. You wipe with some the alcohol, or you sterilize that one with the high temperature provided in the incubator. Make sure that that works fine for others also, because when too many people are using, naturally the chances of the contamination will be a bit higher. And sometime if you forget to close the cap of the flask, that it may spill something and it may contaminate others' work or even your own work. That you have to be careful. And the other thing was that I already told what are actually adherent and suspension cultures. Adherent is the one where it is attached to the surface, and the suspension is the one which is not attached to the surface. So why fetal bovine serum is commonly used in cell culture media? So this is actually there is so many studies that are going on. So fetal serum, this fetal bovine serum is actually a costly one. So sometimes you may have to add it, or sometimes by adding itself you are creating the problem. That's why here fetal bovine serum, it is rich with various nutrients that are necessary for the cells to grow. Okay. So if you provide, maybe there are certain media you are supposed to provide that fetal bovine serum for the growth, like DME medium, you have to provide 10% FBS, or for that matter you may have to use serum regularly for the proper growth of the cell. It depends on the type of the cell what you are using, and type of the media what you are using. Because the major function of the serum is that serum will contain the nutrients, and that nutrients may favor the growth of the cells, but it should not hold good for all. Some cells may not grow properly when you supplement with the FBS. In such occasion, there are certain serum free medias available, and you can go ahead with those kinds of serum free media. Or here majorly sometimes there are some disadvantages of the FBS also, because you cannot maintain the batches, this homogeneity of the batches. You get the serum from the supplier in one batch, and the same batch might not be supplied by the supplier next time. So you cannot maintain or control the experiment properly. That's why you have to make sure that whenever you are using this serum, you should aware that whether it is giving the proper uniform growth of the cells. And at the same time, sometimes even the presence of the serum also affects your studies. That's what I was telling previously. Whenever you are actually going for the detaching the cells, or we call that word as passaging, where you are subculturing, in that time, where suppose if there is a small quantity of trypsin, it will actually, a small quantity of serum, it prevents the action of the trypsin. The best way is that you wash the cells properly before going for that one. And how does an incubator help maintain the optimal cell growth conditions? That's what I was telling. The temperature, this incubator provides, because this carbon dioxide incubator and the incubator I am going in line with line, where the majorly we are using carbon dioxide incubator while growing the cell, but in certain conditions you may have to have maybe the normally what you are using the incubator without any carbon dioxide supply, where you are setting certain temperature and you want to set the reactions, and that can be done there, and that's where you will get help from this incubator. And how what are the key aseptic techniques required to prevent contamination in cell culture? What are the aseptic techniques? In the sense, that's what actually the good laboratory practice, what I am telling. So when you enter the lab, make sure that you are wearing the lab coat. And once you enter the lab, make sure that you are tying your hair. Okay? Because once you are working in the biosafety cabinet, so that may lead to the contamination of the cells. Okay? Or along with that one, regularly swab your hands with the alcohol, and sometimes what happens, you take the media, and that media you take it into the biosafety cabinet for doing the work. You have taken the media, inside the media it is actually sterile, but the surface may not be sterile. So that will be the source of the contamination. So what do? You properly wipe the culture bottle properly with 70% alcohol, and make sure that keeping in the biosafety cabinet. Okay. And at the same time, whenever you are in the biosafety cabinet, make sure that you are not talking, and your hand should be at least inside about 4 inches inside, so that it will be properly inside that biosafety cabinet, and you are working, and you are not disturbing the movement of the air. And earlier days, the biosafety cabinets used to be provided with the burners. Usually whenever we were transferring the media, the transferring used to be carried in presence of the flames. But nowadays the biosafety cabinets are fitted in such a way that usually we avoid the burners, and you should have a proper arrangement of the materials what you are using. When you go to the biosafety cabinet, you keep the things what is required. Sometimes what happens, we have the habit of thinking that we assume that we need so many things and we keep everything piled up in the biosafety cabinet and you will not able to work. And sometimes whenever you are using the pipettes, the major mistakes what we do, we touch here and there. That may get the contamination. Or even make sure that whenever you are sitting also, you are sitting in a proper posture, and at the same time you make sure that whenever there are certain biosafety cabinets, they are coming up with certain sheets so that your breath will not reach the incubator. That you have to make sure. And whenever you are handling any instrument or any flask, because media you have to transfer. Best way to transfer the media is that either you take the dispenser, either you take some pipettes or you transfer it. Suppose if you have to transfer the media using the bottles only, best way is that after transferring, better you wipe that the bottle properly. Otherwise what happens, that it spills around the rim of the bottle, and that itself, it is a contamination. Okay, so that care you have to take. And so one thing is that another one, what is confluency and why should cell be passage 70 to 80% confluency. Confluency in the cells, when you grow the cells on the culture flask, they will actually maybe they grow, and if they grow 100%, they may start getting detached. Okay, so that may not give you a proper results when you are going for this subculturing, or we call it as passaging. So what best way is that you take the culture media with the cells, you observe under inverted microscope, and then make sure that it reaches only around 70 to 80%. So once it reaches that maybe the stage, then immediately you go for passaging, that is subculturing of the cell. And by that way, what you are doing is that you are avoiding the losing of the cells. Cells, they will start getting detached if the number of the cells, they become increased. And what steps are involved in subculturing or passaging cells? And here, when you are passaging or subculturing the cells, subculturing the cells, what you have to do is that, so what you will do, you will first take the culture and you keep all the things and you follow the precautions whatever I was telling, and then you take out the culture flask in which the cells are growing, and you keep on the this, and make sure that whenever you are doing such things, you keep the laminar airflow. It should be free from whatever that is not required. Make sure that you are keeping that maybe trypsin solution or media whatever you required, or the pipettes whatever you require, everything keep it ready. And then you take the cells and then decant that media whatever that is already present in that culture flask, and wash the cells with the PBS, so that if you wash that one, it is easy to treat with the trypsin. So you add the trypsin, and when you take the culture flask and if you just add it, there is a chance that only certain regions they are reacting with the trypsin. Make sure that the whole area is covered with the trypsin, even if you are adding around maybe 0.25 ml or whatever may be, depending on the culture flask size, and then make sure that it is completely covered that cells, and you wait till the cells they start getting detached, and then you add the media, because here the advantage is that if the media contains the serum can, this FBS itself can act as a stopping agent for the trypsin. So once the cells are detached, you think that the cells are detached, then you add this FBS and block this the further reactions, and you make the suspension of that the cells, and then you have to dilute it. So dilution in the sense, maybe you have got one flask of cell, and that one flask of cell should be divided either 1:2 or 1:3, 1:4, depending on the type of the cell and type of its multiplication capacity. So if the cells are dividing very fast, and if you are giving only 1:2 dilution, it will grow very much faster, and you will not have much time to, you should only focus on passaging the cells, no other work will be left out. And once you do that one, and before doing that, you should know how much cells are present in that the suspended one. You take out and find out how many cells are there. Accordingly, you find out how many the culture tubes in which you have to seed the cell, and then you once you subculture them, keep it in the CO2 incubator, and that day work is done, and clean your that area properly. Make sure that you switch on the UV light for 15 to 20 minutes, and you keep it at in the CO2 incubator, and next day morning you have to come and observe what is happening, because if there is any problem, cells they may not attach to the surface, or there may be some problems, or sometimes the cells may not be detached properly. You find out what is happening in that time, and accordingly you have to design. Sometimes what happens, the cells will not detach only, and that time you have to increase the trypsin concentration, or sometimes the cells will not grow only, maybe because you have added too much amount of trypsin, or you have kept it for long time for the action of the trypsin. So once you can analyze after the next day, once you come, you will get an idea whether you have done it properly or not. This is what you can do. Other things you should be aware that whenever you are doing is that contaminations which you cannot able to recognize, or there are contaminations which you are able to recognize. The contaminations from the bacteria and fungi, it is very easy to recognize, because immediately what happens, the media is added with the phenol red. That phenol red is having a color, and as the pH reduces, that color becomes actually yellow. If you are getting yellow color or turbidity in the media, immediately you can suspect that something happened in that culture. So immediately you better check for the turbidity as well as the color of the media. So, or sometimes there are certain contaminations, it is very difficult to detect. That's why when you are purchasing the cells itself, you should be very much careful. The major problem with the contamination is that mycoplasma contamination. They are the bacterial type of cells without the membrane, and they grow in the media, and they are not acted upon by even the antibiotics. And actually they contaminate almost all the maybe the cells once it is infected in the lab. And moreover, they cannot be visualized under microscope also. So there are different methods that are available. By that one only we can able to detect their presence. Either we have to go from culturing the mycoplasma, or you have to go for 16S rRNA sequencing, whereby you can find out the presence of the mycoplasma. But the thing is that they don't affect too much your the cell culture. You will not able to recognize, but the result what you are supposed to get may get altered because of those organisms. That's why you have to be very careful about the mycoplasma contamination, and that's why the best way is that you make sure that when you purchase the cells, always make sure that you are purchasing from a proper supplier. So authentic suppliers, those who maintain the kind of the American type of culture system discipline or something like that, they mention we are following this method to maintain the cell culture. That would be the best, because here our regular source for the cell culture is the NCCS Pune. So they are maintaining many cells that are regularly used for our studies. So those can be used, and you are sure that they have done all the studies. And if you want, if you suspect there is problem with the contamination of mycoplasma or something, best way is that you can send the sample, because if you don't have the facility, better you send the sample to NCCS only, get it checked, and they do the procedure and they will find out, they will tell you whether you have done is proper or not. And at the same time, always try to avoid, that is the best way. The prevention is better than cure, isn't it? In that way, we have to avoid the contamination. And in between, I did not mention one thing. So usually we do the cell culture, either primary cell culture or the cell lines. Primary cultures are actually, it is straight away derived from the tissues and we grow them. Whereas cell lines are the one, they continuously maintain that line of the cells, that maybe when we are in the laboratory you can see the cell lines. We are not maintaining the primary cultures. Usually whenever we are studying, because in Nag's lab they used to do even that one, because pancreas tissue they used to take and they used to grow also, I have seen that one. If you are working with the cell culture, here we can work, or even you can do that one also. If your work, if it demands, you can do that one also. So that's about the tutorial, and if you have any other doubts also, we can discuss. So as we are running out of time, still if you have any time, doubts, we can discuss at the lunch time and free time. Thank you.
Okay, we are almost there for the day. So we thought just to, according to your schedule, we have this in-vivo cancer models. Okay, I thought even though we have covered most of these cancer models, I think we should just go through and make sure that you know all those models, and also in each one what is the difference, right, and why you want to use it. I think it's important. These in vivo models are essential for studying cancer biology and therapeutic efficacy, and also metastasis in the whole organism. So you all know already in each one, like for example tumor biology, cancer progression, what models we have used, genetic model or any model, cell model or anything. therapeutic efficacy, you know already orthotopic or xenograft and genetic mouse model, why we use it at what stage, what for. And you also know metastasis model, a little bit of it, not completely, but still you know the models, why we use it. And when they metastasize, and also you know, the cancer has to be progressed to metastasize, right? And also, these models, in vivo models, help in validating the in vitro findings. I think I mentioned yesterday that what in vitro findings you have found, and basically you're validating it. For example, I think I was telling someone that you have to see the colony formation assay and see what happens in the colony formation assay. If you have seen the result in colony formation assay, usually it mimics in the in vivo mostly, maybe 90%, don't know, but it will usually mimic in vivo. So if you are doing it right there, it actually in vivo you get that result, unless until there is other technical issues or cells have an issue or anything else. So why we use these models? Because some of these models recapitulate the way the human biology or human cancer progression happens. That's why we use it. That's critical for advancing the translational, of the cancer progression or cancer research, improving also the clinical outcome of this, study, whatever that you are trying to establish or do. So the types of in vivo models, right? We have a xenograft models, we have a syngenic model, genetic mouse models that we talked about. We also talked about patient-derived xenografts, okay? And also we talked about organoids. Those are kind of, we actually derived from the xenografts. It's okay if you cannot do it. At least you have that knowledge of what models you have, we have, I know people have it. Then at least you'll know when to use it or how to use it, right, if we have that opportunity. So in the xenograft model, we implant human cell lines, right, or the tissues into the immunocompromised mice, right, because you are using the human cell lines, the mice are immune compromised, so you basically, you're just testing them, that's all. So you basically not mimicking the whole microenvironment or the whole thing that in how it is in the human, in the mice. Then you use a syngenic mice model where you implant the murine cancer cells, like mice cancer cells back to the immunocompetent mice, where you basically get the immune whatever the microenvironment in those mice. So most importantly, the engineered mice, okay, the spontaneous tumors are fantastic because it has everything, how you see in the human, right? Probably immune microenvironment, all other things. That's why, that's the reason that people try to establish in the genetically engineered mouse model something if they want to study either biology or therapeutically, right? Patient-derived xenografts are very important nowadays. If you think the technology is so much developed, but you all know probably that the patient will be lying on the bed and they take the tumor and we will be working in the laboratory. For example, our own laboratory, what we do is we take the patient tumor and we divide the tumor and we study everything. Like one person will be studying something, one person will be cutting those tumors and putting into the mice, orthotopically and growing them, and then again they take and then cut and again they grow in more mice, and then therapeutically they'll be doing it. The patient will be lying, they'll be going through the chemotherapy after the surgery, right? And we do the lot of chemotherapeutic agents and combination whatever we think is promising. And if we see any great result, probably we will take that result back to the clinic and tell them that this is what I think this patient tissue is responding for this kind of drug. I'm just giving you a hint, that this kind of studies are going on in many laboratories and cancer centers all over. So it's also important. Your cancer centers has to have the approval to conduct this kind of studies. Otherwise, not all the cancer centers can do. Okay. And when you take the xenograft models, there are cell line derived xenograft models. Okay, there are advantages where you can establish a tumor, and there is a reproducibility. Okay, is easy. There is a limitation, lack of tumor heterogeneity. Okay, you don't see the heterogeneity in the cell line derived tumor. For example, I was telling you about the human cell line. You take and there is no heterogeneity there. Okay, you just study drug. Okay, the tumor going down. That's it. That's why if you submit any paper, probably I don't know who has any experience, but if you submit a paper, reviewers ask, okay, there is no tumor microenvironment in this. Only you're just showing the tumor reduction. That's not enough. Okay. So the patient-derived xenografts, it's better, it's preserved, tumor architecture, heterogeneity, and genetic profile is there because you're taking the patient tumor and genetic, whatever the genetic is, profiling in the tumor, is still there. But the problem in this study is that you need the immunocompromised host variable, so that's expensive. You have to make the mice with immune, and then you have to study the human tissue in those immune, because you have to develop those kind of mice. It's very expensive. Okay, so I showed you one of the data. First talk, I showed you humanized mice we developed, and then we studied the CREB inhibitor in those because our study took us there, and we did study those mice, but one mice is $3,000. Okay, just imagine, convert into rupees, like how expensive it is, just one mice. So like that, you have to have multiple mice to study. So it's a little bit expensive stuff. Okay, when you take the syngenic models, right? You take the murine cancer cell lines and implant into genetically identical mice. So the advantage is good. It has an intact immune system and allows the immunotherapy studies. You can do, I showed you one of the immunotherapy study that what we did, and that's exactly what I'm trying to tell you, advantage, fast, effective, things like that. But there is, any model you take, there is always a limitation. These models have, they don't recapitulate the human tumor biology. True. It's like, there is no 100% perfect human, model that we can actually study. It's all, but these models will give you some kind of data. That's why you go to the phase 1 and study the toxicity and other things. But it's okay. But important for you is that when you're doing the research or conducting the experiments, I think the thinking all about these things and conducting whatever you can, in a right model that based on what you are thinking, if you can do. If you cannot do, there's always a collaboration. If you cannot do collaboration, just whatever you have, publish it, right? Wherever you can, right? Don't waste those data if that is reliable, good data, okay? So when you take a genetically engineered mouse model, it doesn't matter which cancer. We already discussed that you have a genetically engineered mouse model. They spontaneously develop tumors, and also it relapse and resistance and all those things. We can study a lot of things. There's like you can basic cancer research you can actually study. I told you what we can study is that you take the we did that. We have a KRAS mutant mice model. We have a KRAS and also P53 mutant mice model. In the KRAS P53 mutant mice model, it actually in that develops cancer. KRAS don't develop cancer, but it has KRAS mutation. But it needs another thing. That's why we did the alcohol and induce other things. So we made the cancer. But here we do the cancer research. Like for example, study the cancer progression. You can use those models. Also, we can do therapeutic. You use those models and study because it recapitulates exactly how the human biology and cancer progression happens, right? That's why there is an advantage and disadvantage. There are more advantages than the disadvantages, but it's just that how why you want to study in genetically engineered mouse model is a key factor, right? The limitations are the longer latency periods, higher costs. Genetic mouse model is not easy because you have to maintain the facility and maintain the mice. I don't know whether anybody is working on genetic mice model. It's hard because for the for facility you have to have in the universities or institutions. But those who are having it, definitely there is great papers are coming out from their laboratory. and the complexity in breeding and genetic you have to have the genetic person all the time with you. Because I have a colleague called Michael Vanson. He's always with me, even from Vanderbilt University to now. Now we are a postdoc together, and we went to a faculty, and now he's associate professor in somewhere else, and now I'm in some other university. But he's a geneticist all the time. Anything genetic problem, or I'm not a geneticist, right? So basically, he's the one. So any mice model or anything like that, I always ask questions, and he's my collaborator. We publish together, and we develop anything ideas, and he makes the model, and I actually use it, and sometimes we collaborate each other and makes the model. Like, if I have scientific questions, and I discuss with him and make those models, and we find out, we figure it out, and that's how we develop the relationship in the scientific world and collaborate each other and develop. He's from different Kansas University, I'm in Miami University, it's still like that. What I'm saying is that you have to have someone who understands the genetics of these mice, and the crossing, breeding, and all those things. It's not that everybody, you cannot think like, I can start doing it, but you have to have that basic knowledge, and also, understanding the genetics, which is different. Okay, we have only, if I am from biochemistry, I studied the biochemistry, a little bit of genetics, a little bit of molecular biology, a little bit of microbiology, immunology, stuff like that, but not in depth anything, right? So that's why you need to have the genetics person. So if you are developing a mouse model, it is a zoology, like where they actually understand the chromosomal mice and those kind of things. They can actually collaborate with zoology people, those who are expert in genetics, something like that. So when to consider the model selection, right? There's a great website here, Cancer Model Database from the Charles River. And you can actually go there and check each model that you want to study, and they'll give you all the details about each model, what these models are, and like that. There are even in NIH website they have it, all the details about models. But what I'm saying, even the Jackson laboratory, they have the mice model and the details. So like that, there are you have to spend time to figure it out, all these models, and if you want to. Like, if you don't want to, it's okay. But at least you get a knowledge. You can talk about it, and you can discuss with someone and get a knowledge of the models, right? Does the model mimic human disease features? There is a question. You have to always have that, to relate your biology to your model, and also there are like an immune competence with immune system. Do you want immune system in your mice model? Otherwise, why you spend so much of money? You don't have to. Think about your model, think what you are working on, and then you think about whether you want immune system or not. I gave you an example. In our study, we wanted immune system because it did not work MEK and STAT3. And then we looked at all the immune population. Then we found that, yes, we need the immunotherapy. Then only we went for immune related studies on genetic mouse model, and immunocompetent, immunized mice model, things like that. So it all depends on your scientific question, and also what type of data you are getting it from your research. Tumor heterogeneity, again, you have to think about, do you want to study heterogeneity in your model? Why? If you're just studying the natural compound, just study the way that my first paper was, that we did not do much, okay? But study and see what's going on. And then you move on we moved on to the immunotherapy later on. Like we took 9 years to 8 years to think about it and see. Meanwhile, we published other papers. What I'm saying is that it takes time, and also you have to have the money and all those things. So you have to think accordingly. Okay. The cost, practical consideration, cost, time, and technical feasibility, whether you have that expertise and things like that. Okay. Experimental design consideration. So controls, replicates, how many mice you want to have. Now, let me tell you, 3-5 mice is not enough if I want to publish in Cancer Research Journal. Cancer Research Journal I published before in 15 years ago, and they were fine. 3 mice, I published it. And now it's not enough. So now they want more. 10 is at least minimum sometimes, because of the data, that what type of data you are generating. You know what I'm saying? So it's all about that. So the control, because the replicates, the reproducibility issue, like if you have a 3 mice and you're like that, like, whatever is happening, and if someone wants to reproduce your data, it's going to be difficult, right? I think that's the reason that they are asking more mice, right? Important. It's just, even though they don't ask you, they reject your paper. They won't tell you why they are rejecting also specifically, right? There may be reasons for that, okay? One of these may be the mice number. The endpoint selection, what are you trying to establish in your study? Only tumor growth, metastasis, survival? Those kind of questions are also important when you are designing your, in vivo models, okay? Based on that, you have to design your in vivo models. You don't have to go, design or what you want to do, I want to design. There should be your question or your data should take you there. And also ethical consideration, animal welfare guidelines, and also minimizing the suffering. So there is always a guideline for everything. Probably for the human patient, you can actually cut and open, easily, but mice, you cannot do it. You have a lot of restrictions. I don't know here, but in US, you cannot just handle the mice like how you handle human. Human is easy compared to mice. Let me tell you, there is a lot of rules and regulations. Even to each mice that I have to sack it, I have to have the regulation, each and everything, how exactly I'm going to sack it. Whether I'm giving the pain to the mice or not, and how long I'm doing the drug treatment, what food, and whether the light is there or not, whether they are sleeping or not, whether they are active or not, whether they are having any problem or not. I don't think we care about so many things like in the OR also, in the surgery. I don't think so. So it's like that. So we have to take care of, like, for example, I got 3 emails from an animal person that something is happening in some of my mice, that somebody has to go and take care immediately. You know what I'm saying? So it's like that. Anything happens to any mice, like immediately someone has to go and check, make sure they are fine. So it's like that. So what I'm saying is that ethical consideration as you have to consider. So there are challenges and limitations. Of course, when you start conducting these experiments, you will know. Okay, it looks fine. When you see the data, it looks fantastic, everything. But we do go through a lot every day. Okay, as I said, ethical considerations are very strong ethical considerations in US. So basically, we have to go through a lot of things. The differences between the mice and the human biology can affect the translatability. Yes, not all your data, not all your research translatable. Not all. Okay, some fail. Many reasons. Each reason is important reason if you can figure it out. Okay, important for the scientific community. It doesn't matter what type of limitations or problems that you have. Each problem is important. Anything that you think your data is correct, that means it is important to the scientific community. So microenvironment, right? Any difficulties in replicating the human tumor microenvironment? Yes. You cannot replicate the tumor microenvironment from human to the mice to any other model that you think about. We think we are just conducting is, we just don't know a lot. Okay, we predict a lot, and maybe technology is there, but technology is there, we are using it, and we think that that is how it is working. Probably we might not know a lot. Okay, we might know a little bit of it, what's going on in these mice. Okay, that's how probably we also scientists say, in cell, we understood only a little bit of it. There's so much that we don't understand. Okay, so like that. So the same way in the microenvironment, we don't understand so much. But important is that we don't have to worry about the negativity of what is the problem. We have to think about the positive. Like we have these excellent mouse models we should study. We should be able to establish some research and science, right? That's what you need to think about. Don't worry about the limitations a lot, but you need to know. Okay. The cost and time for developing the GEM and the PDXs is a lot. You have to have a lot of money to do all these kind of studies. But if you have money, I know there are a lot of companies around in Bangalore. I know they make the PDXs GEM model from the companies, and I know there are some few companies came to AACR meetings also from here, from India. So also these companies are selling all these mice and the PDX. If you have money, I think you can actually get those mice and study. So the best practices. I mean, I think Joshi was talking about in vitro study. Here also in vivo, the standardized protocols are there, you have to follow, and then validating the model relevance to the human disease always. Why are you studying in vivo? Because you want to give the relevance to the human biology, human disease. That's why you're studying in vivo. So that's what I think you need to think about what you are validating is whether it's helping you, whether you're actually getting the data or not. So the future direction, I don't have to say future direction, but actually it is true that you need to develop the humanized mice model to incorporate the human immune component. It's important. Okay, when you're studying, you have to think about it. When you're writing a project, when you're proposing projects, right? Okay, you have to propose something innovative, something proposed, and see how much money is involved. Talk to company and get those codes and add it, and think about it whether you have the facility. If not, if you don't have it, attached to someone who has the facility, maybe from here to Manipal. Manipal maybe having all the fantastic facility, collaborate with them and try to establish your model system so that they'll fund you, they will give you the money. If your science is good, they will definitely fund you. If your science is, like your thinking process is better than anybody else, definitely they'll fund you. It's just about your process. Okay, if you say you're not getting funded, that means that I will say your science, your thinking is weaker. I think you have to become stronger. You have to think beyond. That's why you're weaker. So you have to think how your science can be innovative, right? So the real-time monitoring of tumor dynamics, advancing imaging techniques like PET, CT. I've showed you some of those images, mass spec analysis. What I did not show, I showed you a little bit of those figures, MRI analysis. And what I did not show you is that the whole process of imaging technology, because that's another research and another area. We have done a lot of in vivo imaging, PET, CT, MRI designing and even throwing to the mice and getting those data and seeing all this data, entirely different area. Okay, we did not touch those. Okay, multi-omics integration. So how you can increase? We already talked about genomics, proteomics, transcriptomics, why, what, and all those things. I think it's all about whether how you are doing in the in vivo model. In vivo model, like how you do the in vitro model, it is not easy. Again, in vivo, your data will be different. So you have to think a lot. There is a lot of research. That's why it takes a lot of many years. It's just not 1 year, 2 year, 3 year, 4 year. It takes years and years. The reason is that there is a lot to understand and study and things like that. So your whole life will be thinking about a lot of these technologies and ideas. Even you say something, right? Like your 10, 15 years will be gone for studying just for that. That's why scientists are lying somewhere in the corner and they keep doing what they want to do, but they don't care about anything else because that's what it is. That is the passion that developed, right? Take home message: it is all integrated, well-designed in vivo model strategy is critical for advancing the cancer research and improving the patient care. Very much. In vivo data is a key thing. You might have got a lot of in vitro data. What are you going to do with it, right? What are you going to do with it? You have to somewhere, like you when you come to in vivo data system, in vivo data, again there are a lot of problems. Yes, there are problems. There is always problem everywhere in the research, and when you go to, but you should not think about it. What you need to think about it, whether your science you are translating it or not. If you are in the cancer biology, it all depends on your disease and what type of in food technology probably you think differently, right? Your ideas are different, and your improvement and what you the best food product or best something, and how you package it and how you sell it, how you design and packaging system or freeze drying, or how did you dry, or whether you're keeping all the flavor, keeping all the protein intact or not, things like that. They're different, but it's the same thing. End of the day, we all talking about the technology. We are talking about system, and to keep going in the right direction and making the right path. Okay. Thank you so much. If you have one question I can take, because I think it's the lunch time. You all hungry? Any one question? So if not, we can discuss during lunchtime. Okay. Thank you so much.